Broadcasting live from SETI Alpha 6 in the Andromeda Galaxy, this is Mr. C.
Bonjour mes amis, comment ça va? That means good day my friends, how's it going? Well, number of do nows, so if you'll take care of all of these, of course, at the top of the list, homework in lesson planner, you see it up there on the board regarding LS3. So if you'll stick that in your planner, I'm going to slow down as you do these, make sure that you do these do nows. The worksheet, chemical compounds in cells, needs to come out along with a red pen. We will grade that in a little bit. If you'll also take out LS2, I want to make sure that you and I are on the same page as far as Friday's test. A test, you say? Yes. It could, let's see, I'm trying to think. It might be the last test of the quarter. So I would plan on studying really well and doing really well. So it'll be one of the last big opportunities for you to firm up your first quarter science grade. So I'm assuming you've got homework in your lesson planner now. So let's look at LS2 for a little bit. I might just tell you really quick, and you actually could stick this in your lesson planner. Now here it is, just Tuesday, right? And I'm telling you in general terms what's going to be on the test Friday. LS2, 3, and probably 4. The key concepts. And we're going to review LS2 in just a little bit. We're not going to take very long, but have LS2 at the ready. And you're going to make sure that, yep, got that on LS2. The microscope parts of a microscope will be on Friday's test, and procedures, and then Psalm 139, 13 and 14, you'll be writing it. For you formed my inward parts, you knitted me together in my mother's womb. I praise you, for I am fearfully and wonderfully made. Wonderful are your works, and my soul knows it full well. And the wonderful work there, I'll probably ask that question, refers to us. Us. Okay. So anyway, that's what's on Friday's test. Okay, LS2. Here it is. Looking inside cells. LS2. I've got mine all marked up. Yours needs to be marked up. So you've got it in front of you. You might use your red pen on it. We've already marked this up some. I know we have. But I'm just double checking that you've got everything highlighted that I tell you about. One of the first things I'm going to ask you about, not necessarily one of the first questions on the test, but it's this right here, that every part of a cell is designed with a specific function. Now it doesn't say designed there, but it might as well. It's a God thing. Each one of these organelles has a specific structure so it can do specific functions or jobs. So that key idea right there. And then what do we call these smallest parts of living things? Smallest parts of plants, animals, and humans. Cells. You remember Robert Hooke noticed them in plant cells. Now the ones he looked at weren't alive, but he still noticed them and gets credit for their name. Are you going to ask us, what is Robert Hooke famous for? No, I won't. But it'd be kind of cool to remember that, I think. Anyway. And then you've got the outside border of a human or animal cell, the membrane, which only lets certain things in and keeps other things out. Nope, you ain't coming in. No. God designed them to filter, to be kind of a filter about what is allowed into the cell and what is not allowed into the cell. So the membrane. Then I want you to know the term, what are these individual complex structures called? It's right there, right in the middle, the top box of LS2, organelles. So make sure you're highlighting it. Why would you not? Pay really close attention right now. Why wouldn't you? And make sure you've got all of these down. Yep, got that. Part of your homework, after we're all finished, and while you're waiting to use a microscope, is going to be review LS2. So then you have the liquidy part inside the cell. It was on last week's test. It's kind of a jello thick liquidy part that these organelles are found in. That liquidy part is called the cytoplasm. And then you have the control center of the cell that runs the cell's workings, directs the activities of the cell. Nucleus, of course. So membrane, cytoplasm, nucleus. Say those three to yourself, not out loud, but do the hand motions. Go ahead, do it right there. Yeah. Mr. Gilbert's watching. Do it. Membrane, like the filter. Cytoplasm that the organelles are in, and the nucleus that the DNA is in. Okay. Got to know those. Then you've got to know the next one is a lysosome. I don't think we crossed that out. Lysosome. You see it there right underneath nucleus. You know, I've been trying to think of a way to help you remember what the lysosome does. Lysosomes in cells break down food particles into smaller particles that the cell can use. So you know what I was thinking? Slice, like cut up, sosome. Slice, lysosomes. I don't know, I'm trying it out. I don't think I've ever used that hint before, that learning hint. Slice, sosomes. They slice or break down food particles into smaller parts. So there you have lysosomes. I tried my best to help you remember slice, sosomes, lysosomes. And then you've got the mighty mitochondria that change the energy in the food you eat, munch, into energy the cell can use. So they change it. That is so complex. All of these. I wrote down right here on the side, you see where I wrote, wow. Because all of these parts are amazing. All these organelles. Could they have ever happened by accident? Oh, come on. I mean, it's silly to even think that. And yet evolutionists think that. Just ridiculous. These all are so complex. Just the level of complexity is so high on all of these organelles. So the mighty mitochondria, and then at the top on the right, ribosomes, RBP. Ribosomes build proteins. RBP. Ribosomes build proteins. Do two of them. Not too loud on the table. Not too loud. Don't knock your plastic divider over. RBP. Ribosomes build proteins. RBP. Ribosomes build proteins. So you've got the ribosomes, and you have the vacuoles. Starts with a V. And I'm trying to do it kind of like a container that stores things. Whoop. Waste, water, whoop, stored in the vacuoles with a V. You see it there. Then you have the two parts that plants have, green plants. Thick outside cell wall. And then the greenish chloroplasts inside that have chlorophyll in them. Now I want you to add right here at the bottom. Look how I added it right there. Chlorophyll in the chloroplasts. That's the green pigment. And when light hits it, it makes energy for the plant. So chlorophyll, light strikes it. Again, the level of complexity, over the top. So light hits the chlorophyll and turns it into sugars, energy, that the cells can use to stay alive. Now there's a term for the process. I do want you to put it down. I don't know if I can get it close enough so you can get it in there. Let's see if I can. No, I don't think so. It's photosynthesis. So you'll want to put it down right beside chlorophyll. Photosynthesis. Photo, P-H-O-T-O, which means light, and then synthesis. Synthesis means to put together or make. So what's the idea there? To make, using light, energy for the plant. Photosynthesis. You've got it down. I put it right down below chloroplasts. You've got chlorophyll and then photosynthesis, meaning to make with light or make using light. Okay, down below there are six chunks of information that I want you to know down below in the bottom box of LS2. The first one is an organism, living thing, that is unicellular. Now, even if you didn't know the word, and you might not, you ought to be able to break it down. Uni, meaning one, and then cellular. So the idea of one cell. Unicellular, like protozoa that you're going to look at under the microscope. So highlight in LS2 right there, unicellular. And then multicellular, many cells. There are organisms that have many cells. Can you think of one? You're looking at one. Multicellular organism. You are a multicellular organism. You're made of trillions of cells. So one cell, unicellular, or many cells, multicellular. Okay? Then over on the right side, I want you to highlight or circle organism. Right above organism, right there, I want you to put living thing. Abbreviate living, L-I-V, a living thing. An organism is a living thing. You are a living thing, an organism. So here's the story. In multicellular organisms, and you're going to write this down. I wrote this down at the bottom. See if you can see it. Down at the bottom. Cells, see that at the bottom? Cells are made of tissues, which are made of organs. So you're writing this down at the bottom. Cells make up tissues, which make up organs, which make up systems, which makes up the whole body. Have you got all those down at the bottom there? Fill S2. Cells go together to make sheets of cells. Tissues. Those tissues make up organs. like your lungs. Be a good example. Heart, organs, which in turn makes up organ systems like the respiratory system. Your lungs are a part of the respiratory system that involves breathing, of course, exchange of gases. And then the respiratory system is one system that makes up all the systems of the whole human body. So see if you can say all 5 of those to yourself. Cells make up sheets of cells, tissues, which make up organs, which makes up organ systems, which makes up the whole body. I want you to know those. So that's LS2. So there you've got LS2. Now I want to correct the worksheet. We'll correct it pretty quick. And I want to tell you about your homework. And then I want to turn you loose to work with the Microscope Lab. So, all right, let's do this. Now, you know you needed page numbers. So each one of the right answers is worth 1 point. And then put a check mark right by the page numbers. I'll tell you how to score the page numbers at the end. All right, each correct answer is worth 1 point. Number 1, carbohydrate. What's the answer? C as in Charlie. An energy-rich organic compound. Examples, starches, sugars. All right, number 2 is carbon. What is that? That's D as in delta. Number 2, D as in delta. An element that's part of most organic compounds. Compounds that make up living things. Number 3 is A, alpha, for inorganic compound. Water is not alive. It's not a living compound. It's inorganic. By the way, those first 3, the page number is 24. 24, 24, 24. So put a check mark beside each of those if you had the right page number. Now, if you didn't have the right page number, be honest. You are LO students, be honest. I'm trusting you to be honest. So page 24 on all of those. Hey, I'm giving you the page number and number 4. I'm just giving it to you. Okay. All right, number 5. Now, this is those true and false statements. If it's true, you get the point if you just have true. If it's false, you get half a point. If you have false, you get half a point. Then you know what you've got to do. You've got to correct it. Make it a true statement. All right? Number 5 is false. So half for false. And correcting it, sugars and starches are examples of, I just said it, carbs or carbohydrates. So half and half, 1 point on that. Number 6 is false as well. Proteins are part of cell membranes and store energy. No, not proteins. What are they? What's the correct answer? Lipids. Lipids, lipids. So false for half, lipids for half. Number 7 is true. If you had true, 1 point. Here are the page numbers. Number 5, 24. Page 24. Check mark if you had that. Number 6 and 7, 25. Page 25. So check mark if you have the correct page number. Number 8 now is false. It's page 26. Number 8, it's not carbohydrates that direct cell function. It's nucleic acids like DNA or RNA. So number 8, false for half, nucleic acids for half. Page 26 again. Check if you have the right page number. Number 9 is false as well. Half for false. Water makes up 1/3 of the human body. No. 2/3. So 2/3 to make it correct and get the other half to get the full point. Page 27 for number 9. Check mark by 27 if you had it. And then 25 is true. So if you had true, you get the whole point. Page 25. Okay? Now, if some of you missed something, perhaps Mr. Gilbert caught these. At the top, I'd like you to put your score over 10. Number right that you got correct over 10. Be honest. If you didn't correct some of those falses, then you only get half a point for each of them. So number right over 10 at the top. And then here's what I want you to do on the page numbers. If, I'm going to do it this way today, okay? The page numbers are important. I don't want you guessing. So, if you got at least 5 of the page numbers right, okay? So, at least 5 of them right, that's a good thing. No deduction so far then. Nothing to take off. Now, if you missed two to, I'm going to say, I get this right, 2 to 5 page numbers, you're taking off 1 point. If you miss from 6 to 10, you're taking off 2 points. I want those page numbers. So do the deduction at the top, decide your score, just put -0, -0.5, -1, -1.5. Well, it would be -1 or 2. So -0 or -1 or -2 beside your score over 10. Don't change your score over 10. I'll change it. But you're putting your deduction for the page numbers. Okay, again, if you missed 1 to 5, deduct 1 point -1 beside your score. If you missed 6 to 10 pages, page numbers, -2. Okay, so there's that. Okay, doing good. Homework, here it is. It's on the board. You've got it in your lesson planner. Read and highlight LS3. You should be getting LS3 from Mr. Gilbert. I don't think it's handed out yet. So read and highlight LS3, and then you need, on the white sheet that he's providing, you need 12 neat, complete bullet facts from LS3. 12. Skip lines in between them. 12 neat, complete bullet facts from LS3. Do a nice job on those. Now, I'm going to let you in on a little secret. These worksheets and these bullet facts, it's an easy way, in one way, to pick up points. They're not hard test questions. So don't tell anybody, but it's the way you fatten your average. It's the way you raise your average by doing well on this daily work. Worksheets, bullet facts. Just saying. Don't tell anybody. Okay? And then the second part of your homework is to RR LS2 at least one time. Go through LS2 and all the key concepts and their meanings. I'd say at least 5 to 6 minutes. Go over it, minimum. At least 5 to 6 minutes going over LS2. Okay? So I added the LS2 part. I don't think you're finding that up on the board. Now the Microscope Lab. Here's the way it's going to work. In your journal, you're going to dedicate a page or two to the Microscope Micro Lab. And then you're going to number 1, skipping a couple of lines, 2, skip a couple of lines, 3, skip a couple of lines in your journals. I'm going slow so you can get the numbering in. You're numbering to 8. There are 8 microscopes with microscope slides on them. I told you yesterday what all the parts would be. I hope you're excited. By the way, have most people who ever lived in history had the opportunity to look through a microscope? Say no. No. You have an amazing opportunity to look through a microscope. Mr. C, how many times have you looked through a microscope? Hundreds? Hundreds? Do you still like it? I do. It's still exciting. There's something wrong with you. No, I'm just curious, and I love nature, and I love learning things and seeing things in nature. So when Mr. Gilbert numbers you off and sends 8 of you to the scopes while the rest of you work on your homework, you are going to carefully take off the dust cover. Don't bump the microscope. Other than taking off the dust cover, don't touch the microscope. Remember, you know you're in the perfect position. Here's the dust cover. You're in the perfect position to see each thing after you turn the light on, click the light switch on, and then slowly move your eyeball in. Did I mention slowly? When you feel your eyelashes on the eyepiece, you're right where you're supposed to be. Don't go in any closer. Slow movements around the eyepiece. And it's already in focus. You should be seeing some really cool things. I have labeled what you're looking at. You label it and then put it in your journal. So I hope to get at least 2 groups of 8 in today, and then the 3rd group tomorrow. So work on your homework. 12, read and highlight LS3, 12 neat complete bullet facts on the white sheet. Use the backside if you need to. It's all right. Okay. And then study LS2 for at least 6 minutes. Study the key things that I wanted you to remember. Oh, all right, 5 minutes. But at least 5 minutes. Okay? All right. Get going. I'll turn you over to Mr. Gilbert.